All right, we're going to get started while a couple other people trickle in. Thank you so much for coming and welcome. My name is Chelsea DiGiuseppe. I'm the marketing manager for UNHInnovation, which is we are housed in this facility. So this year's Wildcatalyst Seminar is launching research-based startups, and so our aim is to help demystify the startup process and explore opportunities, challenges, and the pitfalls of launching a business by hearing about real-life successes from the innovators themselves. So while the general topics will be universal to all entrepreneurial-minded individuals, there is an emphasis on starting business around university research. And we're excited to explore the intersections of entrepreneurship and academia. So before I introduce our speakers, I just wanted to say a very big thank you to our generous sponsors. There's Hamilton Brook Smith Reynolds, and they're a Boston-based full-service intellectual property firm. Finch & Maloney, a Manchester-based law firm that provides intellectual property counseling and services to tech companies. And Saunders & Silverstein, a law firm in Amesbury, Mass. That provides services related to trademarks, copyright, internet software, and entertainment law. Following today's presentation, there's going to be some networking time, so please feel free to stay and enjoy some appetizers and refreshments and continue to talk to our speaker and to each other. Next month, our speaker is Dr. Debarshi Nandi, and he is a professor of finance at Brandeis. He has started a company based on his research, and it's called Cleanfield Capital. And it uses one of his innovative financial models to make the process of cleaning and redeveloping abandoned and potentially contaminated land more efficient and less risky. The company also has a focus on environmental sustainability, so it's going to be a pretty interesting conversation. So that is coming up on February 22nd, so look out for that invitation probably next week. If you are not on our mailing list and would like to hear about the rest of our events this year, there's a sign-up sheet on the table out by the front entrance. All right, so now I'd like to introduce you to our speaker tonight, Dr. Ed Goluch. He's an associate professor of chemical engineering at Northeastern, and he's currently on sabbatical getting his startup off the ground called QSM Diagnostics. The company is developing handheld sensors to detect bacterial infections in animals and humans in minutes and right at the point of care. I've been thinking about it as the Star Trek tricoder. Yeah, I'm sure you hear that a lot. So we're really excited to learn more about his research tonight. Ed has taken advantage of as many different startup and mentorship resources as he could find, it sounds like, at Northeastern and in his surrounding community. So I'm sure he'll have a lot of really valuable experience to share with us tonight. Ed, thank you so much for joining us.
Thank you for having me. Well, it's nice to know that at the very least I can have this opportunity to present what I've gone through and have this as part of my outreach and educational mission. So I'm happy to be here and to talk about the process that I've gone through. So I'll start out with just a little bit of background on how I ended up going down this entrepreneurial and startup path, and then I'll go into highlight a few things. And feel free to interrupt me if you have a question. I'll dive into things a little bit deeper. And this is the first time I'm actually giving a talk without any slides. So for the faculty in the room, ooh and ah. But business people tend not to use slides, or they'll just have some pictures on it and they're mostly talking to keep everyone's attention. So I'm really trying to learn how to be a business person and go through the process this way. So my background is very interdisciplinary. I did all of my degrees at University of Illinois at Urbana-Champaign, and I started out as a chemical engineer for my bachelor's degree. And then I stayed for grad school, which meant I had to switch to different departments. And I got my master's in mechanical engineering and a PhD in bioengineering. My PhD was focused on diagnostics, lab-on-a-chip technologies. And I guess I'll say a little bit about that. So my PhD advisor was very entrepreneurial, and our collaborators were very entrepreneurial. So I worked with a guy named Chad Merkin at Northwestern. Some of you might know him. in the chemistry department there, and he's had multiple successful startups. And so the part of my PhD technology was looking at miniaturizing one of the technologies that they commercialized, although none of mine made it to market, but I was already thinking about that and exposed during my PhD, and ended up having to use some of the company's things and learning about the business process at that point, and realizing I really didn't want to be locked in to business things at the time. So when I finished my PhD, I really wanted to go back to more fundamental science and technologies for the sake of developing new technologies and understanding new concepts. I ended up doing a postdoc in the Netherlands for 2.5 years at Delft University of Technology in the bio-nanoscience department. So we were doing single molecule electrochemistry, and that was really great. But that's when I realized I was an engineer and I wanted to know what I'm going to use this technology for. So I applied for faculty positions, and when you apply for faculty jobs, you need to present what your potential research is going to be. So during my postdoc, I started to look around at, well, what types of things can we use this electrochemistry for? And I came across some technology, some papers that talked about studying bacteria, and particularly studying molecules that bacteria produce that are electrochemically active. And I saw there was a little bit of publications about this, but really not much, and definitely nothing very quantitative. So that was my idea for the research project. So I got the job at Northeastern in 2010, and I started working on making these sensors and putting bacteria on top of them and measuring what types of molecules they produce. And I just wanted to see what they produce and what quantities, how that changes when you change their environment. And that was one of the first NSF proposals I submitted, and it happened to get funded. And so we went down that path for a few years of looking at different bacteria, different species, what other molecules do they make that you can measure. And that was really interesting and fun for me. And when we started publishing our results, one day the university wanted to do a little media blurb on our publication, and the reporter asked me, well, what can you use this technology for? What is this information useful for? And one of the things I was sitting there thinking, I'm like, well, I guess you could make a sensor, and if you have a unique molecule that you can identify, you could have a sensor. And you could do this at the point of care and be able to diagnose infections. And so that was written up in the blurb, and that's where this kind of entrepreneurship story starts. So it was completely random. There's a healthcare consulting company called VIE Consulting out of New Jersey. They happened to read the story and they contacted me. And they asked, is there a product? This seems like a really cool idea. We work with a lot of hospitals that get infections. We'd like to be able to diagnose them faster. And I'm like, there's no product. This is still just a concept in the lab, but it seems to be working pretty well. And they said, well, maybe you should think about commercializing this and making this into a product. So that was the first idea of commercialization for us. Having gone through that as a PhD student, I wasn't super thrilled, but I'm like, if there's an interest, I'll pursue this path and see where it goes. I knew from my PhD training that along the way we should be filing for patent disclosures as we come up with these ideas. So we had already started filing some patent applications on the sensors that we were making because they were so small you could put a single bacterial cell on top of them and see what's happening in real time. So maybe there could be some value to it. And when I had heard from this consulting company that there could be some interest in this, I reached out to Tech Transfer, and obviously as soon as I filed the disclosure, they wanted to know, well, who was going to use this? And I told them, I'm like, well, healthcare companies might be interested in this. This is how I could envision this going forward. So they reached out to companies, brought them in, and we had conversations about licensing the technology, what you could use it for. But I always got the question, so who's going to support it? How is this going to be developed going forward? and I didn't have an answer to that question. I asked my grad students at the time, and they weren't super thrilled. They also like just working in the lab and developing this. So for a while, I didn't do much with it. But then this company and some other companies kept contacting me and asking me, like, well, what's going to happen? Because we had new papers coming out showing more of the data, and I started thinking about this in terms of healthcare application. And at some point, I'm like, okay, let's see if we can commercialize this. Is there really something here? So I asked my tech transfer office, who do I talk to? I have no idea about how to actually make this into a product or start a company. It looks like the companies that are talking to us aren't going to move forward with it on their own. And so they pointed me to two different directions. One is a group we have called IDEA, which is a student-run accelerator program that is open to anyone in the Northeastern University community. And they give you the basics of how to commercialize a technology, build a pitch deck, pitch to people, and they will assign you a mentor and give you kind of a crash course in it. The other group they pointed me toward was health science entrepreneurs. Now this is a more specialized group that we had on campus that only works with biotech. And I contacted them. They sat me down for an interview. And so that group was founded by a set of alumni in the biotech space and healthcare space that have come back and are working with the university. They interviewed me, and they thought there was some value to this technology. So they said, well, we think this is something worthwhile to go forward with. You should think about commercializing it and seeing where this goes. And I didn't know anything about how to start a company and do these things, and that's what they're for. So they said, we can try to get you some mentors, teach you about what you need to do, how to make a pitch deck, how to remove all of the data from your slides and talk about the important things to business people, telling them how much money they can make out of this, where they can use it, and go down those directions. So what they did is they prepped me to do a 5-minute pitch to their mentors. So they had an infrastructure of mentors at the university that would get together and listen to companies talk and figure out how to match this. And they were just starting to formalize this process. And it was going to be called a Venture Mentoring Network. And they would do it as a lunch once a month, and they would have 4 or 5 companies come in, do the presentation, and the people in the audience could mark down if they're interested. And so I was one of the companies at that first lunch that presented what my technology was, what I thought we could use it for. And out of that, we had 5 people sign up that marked down they want to talk to me because they think they can help me with this, and they thought this was an interesting technology. So I got to meet with them, and this mentoring group came together, and HSC would organize us to come in once a month, sit down for an hour, and talk about what we're doing, how to develop a business plan, what would you do to commercialize this, what kind of team do you need. And so that's what we started working on. And around that time, my first PhD student was getting ready to graduate, and he was looking for a job. And he really liked the sensors and said, well, would you be interested in taking over this technology and commercializing it? And he said, well, I don't know anything about it, but seems like an interesting concept. And we learned about the I-Corps program. And we said, well, let's try to do this. You can learn about it. I can learn about it some more. It's a way for me to bring you on as a postdoc, and we could see if we can transition this over. And we needed to have an industrial mentor that would go with us on this I-Corps program, because it's a very intensive 6-week program. And so we didn't have a local node, but you apply at the national level for this. And if your initial concept and idea was funded by the NSF, you can apply for this type of grant. So we did, and we got an interview. And I was asking, in the meantime, all the mentors around who could donate their time to do this. And one of my 5 mentors, Craig Sokol, said he can do this. He had started up companies in the past in diagnostics, in clinical chemistry, and had been successful with it. And he currently had a company that didn't require too much of his time. And he said, this is something really interesting. I think this has potential. I can go with you and help you out on this. So he kind of became our lead mentor for going through the process. We were based out of DC, so we flew down as a team for 3 days. We had to do 15 interviews before we left. So that was a very intense process. They taught us how to interview people that could be interested in the technology. And this was definitely something I'd never thought about before. Usually I'm used to going in and giving seminars at different schools, and I just present my technology and say, here's how awesome it is, and here's what you can use it for. And turns out that is the absolute worst way to interview somebody about your technology, to see whether it would be worth commercializing. Because you get so much bias. The people will come in, and there's a lot of psychology in this. They'll be intimidated. Hey, I'm big shot Professor so-and-so with this technology, and they'll say, oh, this is great. We need this. Of course we need this. And then you leave the room, and they're like, nothing ever happens. Or if you were to ask them, well, pull out and write a check for me, buy one of these, they would probably say, no, I'm not actually going to buy it. So they taught us how to go in, not say anything, and actually they encouraged the students to do this, or said to lie and just say you're a random researcher doing research and trying to learn about what problems people have in a specific field. So we went out and talked to people and did the interviews. And actually, my little anecdote was that while we were in DC, I didn't know the DC area very well. I had to find people to interview. I managed to sneak into George Washington University Hospital. They have a wound care clinic, and I told them I had an appointment, so they would let me through the front door, and I didn't have an appointment. And then when I got to the receptionist's desk in that ward, I told them, like, do you have a nurse or a doctor that's on break that could chat with me? I'm interviewing about some new technologies for potential wound care. And a nurse came out and chatted with me and gave me some answers about it. And then she told her friends, and the next day they asked me to come back. They're like, oh, this doctor can chat with you today about this. And down the road, at the end of our interviews, at the end of the program, we actually put in a prototype in that hospital to do some initial testing for us. And that never would have happened had we not gone on to interview with people. So one of the things we had to figure out, stepping back a little bit, is who's going to buy this technology exactly. So they teach you about this in the I-Corps program, or if you go into business school, you can learn about this over a much longer process. But I loved it as a 6-week crash course to business school intro. You have to identify your market, identify your problem, and what solution you're providing for them. And so you have to segment your market and all these fun terms, because you can't just say hospitals are going to use it or doctors are going to use it. So they forced us to talk to different types of doctors. We talked to surgeons, we talked to pediatric specialists, orthopedics, ICU facilities, people that we thought would use it. And we got a lot of interesting feedback from them in this process. And it was great because we had to do it and get it over with. And we learned a lot of stuff along the way. So coming from an academic background, I read lots of papers, as most faculty do and PhD students. And so I read a lot about surgical site infections. Huge, huge problem. Everyone's developing surgical site infections. So I figured surgeons would love this technology. Our technology, by the way, I think I haven't mentioned it yet. Well, I talked about it a little bit. But what we were hoping to do is to have this sensor sit on top of a wound and check every few hours whether there's an infection starting. Because if there's no bacteria there, you shouldn't see any of the molecules that the bacteria produce. But as soon as you see a certain species of bacteria, they produce this molecule and our signal gets picked up by our sensor. And we would have a panel of these sensors and they would pick up all the common species and send that information back. So with this, somebody post-surgery, you're really worried about the surgical site infection. You want to know as soon as possible when an infection is starting so you can start them on treatment right away. Turns out surgeons did not want this type of technology. They will not take a chance with the patient. They will put them on broad spectrum antibiotics before the surgery, continue it after the surgery. A lot of surgeons leave the wound open. They actually don't put a dressing on it initially. They want it to be in a very clean environment, but allow it to heal under natural conditions. So there's no way to even get the sensor onto the surgical site. So that pivoted us very quickly away from surgeries. Where we did get traction was in wound care. Turns out that's a community that has challenges coming back. They have to see the doctor regularly. And specifically within wound care, we're looking at diabetic foot ulcers. So diabetic community, very large community of people, and about 5% of diabetics develop foot ulcers. And you have to monitor them because they can't see the wounds. Usually they're on the bottom of their foot, and they can't feel the pain because of necrosis from diabetes. So they get an infection and they don't even know it. So if you had a sensor there that could monitor when an infection is starting, they could come in, know to go to the doctor and get help right away. Between visits, we could have the doctor check whether it's getting infected and do things like that. So that kind of helped get us our market focused. One of the mentors we had asked us, well, what about outside of humans? Have you thought about animal infections? So I said, no, we hadn't thought about that at all. Interviewed about 15 veterinarians, and turns out that they're really interested in this. A lot of times pet owners will come in, they're worried their pet has an infection, but they don't want to pay $100, $150 to have a test run and wait 3 days to get the test results back. So if you have a test result that you can have right away while they're still there for, say, the same amount or a little bit less money, they'd probably be more interested in that, and you would get a better diagnosis. And then one of the people we had interviewed was at Angel Animal Hospital in Boston, and they were really interested, and they were willing to said, bring in your technology, we can set up a little study and see how this works. So this was really great. We identified all of these things for having this. Now we had some ideas. We incorporated the company in October of 2014 as a C Corp because we knew that this was going to cost money. We're going to have investors in and figure out how to pitch and how much money was it going to cost and learning all these things. Yeah?
What did you have to benchmark your output to? Or what was the state of the art?
So it's changing, but we're still benchmarking against biochemical analysis. So automated, it's almost like a petri dish, but in 96-well plates, and they run that and they can get their output. So that's what we were benchmarking against. When they got their samples at the veterinary's office, veterinarians at their microbiome lab, they would split it, test our technology and run it on their instrument and see how the results compared. And it'll be similar for human use, although things are going more toward mass spec testing these days. And we're starting to see DNA, but not so much for these types of wounds. You get it for sepsis and things like that.
Those are really far away from sensor technologies, right? Because you need material to distribute into something, right? Wells in a 96-well plate.
Mm-hmm. So our result comes back immediately, and then you wait 24 hours for the instrument to give you the results. Because one of the neat features is while the cells only double, say, every half an hour, they make 10,000 of these molecules per cell every half an hour. So by the time you see symptoms of an infection, the concentrations of these molecules are so high that if you just put a little drop of liquid on there, you can detect it already. So that's what we're comparing against for our technologies. Good question. Yeah?
Was your NSF money STTR or SBIR or what kind of?
That initial one was just a normal NSF grant because I had no intention of doing anything with it. Well, commercially anyway. I just wanted to do it. It was actually a special subset they don't have, unfortunately, anymore called a bridge grant. which was only for the engineering directorate. It was meant to be pre-career proposal because people aren't getting career proposals until their third or fourth year now, fifth year in their academic careers. This was meant to be a first funding with very low preliminary data. Yeah.
And how did you get to Angel Royal? How did you connect with them?
They were down the street from us, and I walked in.
In line with them.
No, that time I didn't lie. I did say that I'm interested in talking to a veterinarian about this, and they took my name and had a veterinarian call me back. And the person that called me back happened to be the work in emergency room veterinarian, the veterinary practice, and she's also head of their antimicrobial stewardship program, which is another thing that's coming up.
So how has the IP worked out with Northeastern?
Yeah, so we were filing IP disclosures along the way. They were turning them into...
Are you happy with the arrangement?
Yes. So we actually officially haven't signed yet. So yeah, let me fast forward a little bit, maybe for sake of time. So we formed the company. We started then negotiating what our IP would look like to license it back from the university, and sat down and got terms. One of the things that unofficially I'll recommend is talk to other companies that have licensed IP and see if they can tell you something about what terms they got to benchmark. There is a very large spread among universities with what they're expecting to get for their licensing terms, and you can kind of negotiate back and forth. I think we got something that's similar to what other startups have gotten out of Northeastern for their license, and we felt pretty good about it. So we haven't signed the final term yet, but one way we got feedback is that I presented that to my mentor groups and to my mentors who had started companies and licensed IP in the past. So they were able to give me some feedback. Oh, this is too high, this is too low. I guess we could live with this. And that mentor group, because they had started companies, also knew angel investors. And so they were able to get me in front of the angel groups to pitch. And so the questions they would ask is, what kind of licensing terms do you have? And they would give me some feedback during the process and say, oh, that seems kind of high, or this is low, or we can live with this. And so that was really good as feedback and leverage on my end from the company side. Hey, I can't get an investor to come on board because this or this doesn't work for them. So that is a really good, I think, way of being able to leverage and work this out. It also helped figure out what fields we want to license the technology in and get that worked out. And Northeastern was nice that they were contacting me along the way and asking me, where would you like to see this IP go? What countries would you like to see it put into if there's a patent issued? And I was able to give them that feedback, both from the academic side and as the potential licensee. So that has been helpful, and I think we've been able to get a really strong IP portfolio that way. So we're going to license 4 patent applications. The first one is about to be issued, and it'll be US and all of the EU. The subsequent ones coming up are US, EU, and Japan, and we feel that works for our market for going through those processes. So a lot of moving parts simultaneously. We were doing that negotiation, we were pitching to investors. In the meantime, the student that wanted to be a postdoc and had started in I-Corps ended up getting a permanent job elsewhere and taking it. So right about a few days before we started, he lets me know that he got this company offer from another company. So I turned to another PhD student in my lab that was continuing the research in this area and asked him, would you be interested in doing this? I think it's a great opportunity for you to learn on the business side of things as an engineer how this would all come together. And he said, yeah, sure, kind of reluctantly to go into it, but it dragged him in a little bit. But he understood what I was trying to sell to him. And he went through the process, so he actually completed this and ended up loving it. So he just graduated last year, loved the process completely, also wasn't interested in working at a startup. So that's how I'm still so involved with the startup in the end, is I haven't been able to get a student to take over for me, but we've had so much interest in this that I've really wanted to keep going with it. And going through all these programs, I've learned all of this information about it. I've gotten really interested and excited about it myself. So we were pitching to investors. There's a lot of other opportunities that pop up. One of them is called Mass Challenge. So we actually applied, I guess, was it last fall? I'm losing track of the dates, but we applied for Mass Challenge and we were accepted as one of the finalists. So we made the top 128. And that was a 4-month program that started during the summer. I'm like, well, I'm not teaching and I'm put in my tenure paperwork already, so might as well try this. And we got accepted, so I was there. Had my mentors on board, and that opened up a whole new set of people to talk to. So that opened up different consulting companies. So we talked to like KPMG, Boston Consulting. We talked to different surgeons at different hospitals and higher ups in administration about this, and they gave me feedback. One of the things my goal was to, at the end of Mass Challenge, was to bring in a CEO, because I can't do all this myself. And the feedback I was getting from angel groups is like, okay, you're a smart guy, we know you can do the technology, but you've never commercialized anything. And especially in the diagnostic space, you have no idea how to go through an FDA process and do this. And it's a really competitive market. So you need to bring in somebody that's going to be more involved that has done this. So with Mass Challenge, I started a CEO search. They helped me put names together. And one of the mentors that I had from the process, so there was another group that had been started up in 2016 called the Sherman Center for Entrepreneurship Engineering. My colleague's going to kill me. He's running this center. But it's just engineering entrepreneurship, and they also have a mentoring program. So similar to what the Health Science Entrepreneurs is doing, this is just for engineers and specialized startups in engineering. So they had partnered me with an alumnus who has his master's and bachelor's in electrical engineering from Northeastern. His name's Phil Devlin. And he had a successful medical diagnostics startup. And when I was doing this search, he said, I'd be interested in becoming CEO. He had sold this company and then left the company that had acquired him. He was working part-time with another company, and he says, I have some time, I can do this. And I thought that was a great fit. I had the other mentors interview him and check that this would make sense. I had him talk to some of the angel groups to see if this is the type of person they would want to see come in as a CEO with the right experience. And they were all happy, so I had him come on board, and he's been our part-time CEO. Very helpful and also complimentary. I interviewed a lot of CEOs that had no science or engineering background, but had a lot of expertise. But they say the number 2 reason startups fail is because they have the wrong people. So this for me was a good fit. He had been in a C-level position in the past, started with a small company that had been acquired and successful, but still had an engineering background, so he could get along day to day and go through these things. So he joined the company just over a year ago. We had applied for some SBIRs right as we were finishing up I-Corps, and we had put it in, but we didn't have all of the information that I-Corps was providing to us, so neither one of those got funded. But then I reapplied last summer, this past summer, again for an SBIR, now knowing kind of what they're looking for and knowing all the things that I-Corps taught me about, because for the NSF I-Corps program application, you need to have some initial business plan in there, an idea of commercialization. Have you identified what the need is, what the product's going to be, what pain points you're addressing? And having that information in there, I think, helped a lot, because we actually got it awarded. So I just got the award letter a few days before Christmas, and so now we're SBIR funded through the NSF for 6 months, and we're developing our pipeline. And turns out for the NSF I-Corps, we actually have to go back, and they have a follow-on I-Corps, so I have to do another 30 interviews, and they look at the next stage of commercialization and help you prep for phase 2 funding with that process. So I'm going to be flying back down to DC at the end of March to keep moving this forward. We've applied now for more SBIRs in the pipeline, knowing that information. I also had a chance to sit on an SBIR panel for NIH, unrelated, but I got to see what other reviewers are looking at. I think we have a much better feel of what that goes in. So I think that's a big portion of being successful at the SBIR grants, is knowing that.
So how much of the pilot data would you suggest is important? Like how much do they value pilot data for SBIR?
I think if you have a valid concept that you can put some meat into as feasible, they probably would go for that. A little bit of preliminary data. You don't need much, especially because they do look on the NSF side, is this translatable? Can this be commercialized? And they weight that fairly heavily. They try to push away from that on the phase 1 at NIH, but the reviewers in the room with me, I'd say 80% were from industry, and that question comes up even though it's not required. They're thinking about that in the back of their mind. Can this move forward? Where is this going to go? And so they value that a lot more. They think you have a PhD, you're working in this area. If you have a pretty good concept, they're willing to give you a phase 1 to see what you can do with it, because it's meant for a prototype. I saw, well, I don't know who's been awarded on the panel I've sat on, but those conversations were coming up, and that's the type of thing they were looking at. Yep.
You mentioned the angels. Have they put any money in, or are they still deciding?
We're getting ready to go back to them now. So we came close to it with a couple of the angel groups. We definitely made it to a deep dive. So after you pitch, if there's enough interest from them, they will set up a team and really vet your technology and the potential for commercialization. And we've made that to that process. And they wanted to see a little bit more data. Now we have the chance to get that data and come back to them. They were willing to put in a smaller initial chunk, but we decided to wait and see if we can get that data. But it was very valuable because you can put in letters of support, and it's encouraged to have letters of support in your SBIR. So I got a letter from one of the angel groups saying that we vetted this technology. If you can get this SBIR and be successful with this, getting this data, we'd be very interested in talking with you and potentially investing. And I think that went a long way as well. As well as a couple strategic partners wrote us letters saying, we think this is interesting, we'd like to see this data. If it comes in, we'll keep talking with you guys. So during that 6 months while we were waiting to hear back, we've actually made a lot of progress with 2 of our strategic partners in negotiating terms. And if we're able to get this preliminary data, that'll move us to an actual investment. So we're on the cusp, and things are moving along with that. And then I went on sabbatical starting this last July, and had the time to really start to dig down into this, start to do some of the experiments on my own. A lot of help from my part-time CEO. The other part-time that he does is actually setting up incubators. So he works for a company called Triple Ring Technologies on the West Coast. It's an engineering consulting firm that has an interesting model. They also have incubator space. So they'll incubate the startups and also do some of the engineering work for them. And now they're trying to do this on the East Coast. So we just got space, and it's called Agility Labs. It's in the South End in Boston. And so we're going to be building out the wet lab space there. But now that we got funding, my company's going to be there in that space, and it worked out really well. So it was a nice balance. I was able to help them with designing the space and bringing in that kind of expertise because they're more electrical mechanical, and bio is a big part here in the East Coast side. So definitely these types of things you start to do as an entrepreneur, and these opportunities that pop up. So I had a friend that introduced me to another startup that needed help with their technology. They got stuck. They were doing sensors. They reached out to me, so I was able to consult for them and help them out with developing their technology. And it's a good way to scramble some resources to keep your company moving. So definitely this kind of bootleg approach. It helps to make up the difference for my salary while I'm on sabbatical. So it's been kind of fun doing that. So I don't know how I'm doing on time. Over? Good? I can talk about other things. Okay. Yeah.
Speaking about, when I think of handheld point-of-care devices, I can't help but think of that epic failure of Theranos.
Yeah.
Did the fact that happened sort of near your launch, did that have an impact with your VCs and your own maybe university contacts? Like, did you get any shaped by like, oh, another handheld?
I did.
You did.
I was surprised at pretty much every person I talk to asks me how I'm different from Theranos. I was surprised, particularly on the investor side, how much people will jump on board. And I think this is another kind of psychology question. Even people on my mentoring team. I've never asked them if they invested, but I feel like some people that I had talked to were investors. And they didn't know what they were investing in. And I had known about the technology. I was reading about it for a while. And from it being in that field, I knew there's something up with it long before they had any questions about it. I had friends at other universities that were questioning this. But you start generating enough momentum, people move forward. And now they're hesitant. They're like, oh, it's another handheld startup that's promising things. But luckily, I have a lot of good answers. I'm like, I actually have a PhD in this. And we publish all of our results. We have patents, IP you can look at. So I think that alone is enough of a differentiator. So there were a lot of marks that they should have noticed on Theranos that they didn't. And that's what I tell them. Hopefully it doesn't affect them. It's still a crowded space. There's a lot of companies. So we differentiate ourselves on the business side because we don't give as much information. So a lot of people have already invested in next-gen sequencing and genomics-based technologies. And they're like, why should we invest in your company? And so it's like, well, you still have to work out a lot of the details for the next-gen sequencing research. They're not going at a point of care. They're still going to be a centralized lab. We're looking at telling actionable information to the doctor while they're still with the patient. We don't give you as much, but we give you something that's useful. And so that's how we're differentiating it. We're very low cost. The R&D is not going to be as expensive in development. So that's our kind of differentiator from the other diagnostics. Yeah.
I'm wondering if your total addressable market's large enough that you could just go to Series A as opposed to going with angels. Cambridge, Minneapolis, and Santa Fran are three areas where I think would invest, and you're right in their backyard down there.
Yeah, hopefully. We've talked to some Series A people. I think it's still tough when you're a first-time entrepreneur. Even with having a CEO on, we need to build out our team a little bit more. We don't have the gravitas of, say, a Bob Langer from MIT that has had proven startup technologies rolled out. So I think if we can get to that point, we'll move. And from some of the feedback we've gotten is that strategics are probably going to move in and partner with us, and we might not even have to, we might go a different route. for doing that. So we're looking at it. I'm also, I won't sugarcoat it, there's still a lot of risk in our technology. Yeah, there's an upside, but we need to develop this panel of tests before we're really set. We're in good shape. All the signs point to it, but the risk is there. Also, the thing that holds people up is that the diagnostic market, the margins aren't the same as a biotech for drug startups. So I just ran into a friend who got funded. Took him a couple years, but he got a big company to come in because he's going to do a new microbiome drug. And just, they don't even know yet what their target molecule is going to be, but the upside on getting a new drug out there is so huge, people are jumping on with investors. So diagnostics has its own little special niche market. We haven't talked to a lot of people in Minneapolis. That's something I think we need to look at a little bit more. The SBIR we got in, I think, will give us the funding and the runway to set up to get us to this next set of data that will really help our evaluation. I should mention the SBIR is actually for veterinary health. So we're making our diagnostic for animal UTIs. And that turns out to be a great business model because you're paying out of pocket. You don't have to worry about getting codes for reimbursement and Medicare and Medicaid and who's going to be the buyer for all this stuff. So it simplifies it a lot. I think that was also a selling point for the reviewers, but we like it as well. And you don't need FDA approval. There's a different path to get to commercialization, and we can piggyback that and develop it. Our people we've talked to have been split on that from the investor point of view. Half of them love it because they can get money back much, much faster with a smaller investment. Half of them think we're too scattered. They want us focused on one thing and don't diverge from the human market. That's never going to be resolved. And so you don't want to just jump in and take money. You want to, I guess, align with the right investors as well that believe in the path you have.
So when potential investors are ready to do their deep dive and they look through due diligence, how much do they come to you and sort of, is it required for you to spoon feed that data, or do they want independent validation of your technology?
They get their own people in. So they want access to all of our publications, unpublished data, anything we can share with them. I have a giant Dropbox folder, and they bring in their own people to take a look at it and give them separate advice.
Do those people communicate with you? They're experts?
In some cases, and others I just got the feedback from the investors. We talked to this person and this person, they like this. They wanted to see how this was going to fit. And
have you ever come across a scenario where they're scientifically wrong based on your?
No, so there is the question mark. So the question with our technology that we're going to get forever, because of how we do the test, is that we detect based on this molecule. And one of the questions that comes up is, how do you know every single bacterial species will have a different molecule? And so far, all the data points that it will. It's hard to prove that everything always will. There's always some level of uncertainty, but we're building up our case as we go along. And there's different fields, and how I started getting into this is actually came from ecology. And quorum sensing was just really brought forth by, I'm blanking on her name, professor at Princeton. She discovered it in squid. The bacteria inside the squid stomach come together and they make these molecules, and when there's enough of them, they start to fluoresce. And from like an environmental ecology point of view, it makes sense that bacteria that can do this and have these unique communications will outperform bacteria that don't, and that there's a need for having these unique molecules. But that's one answer. Question we have to answer. We get the question about antibiotic resistance. Right now, we can't answer that. We have some preliminary data that we think we can address that. Turns out that some of the molecules, because we're quantitative, they get produced at different rates based on whether there's some resistance or not. But even if we don't have the answer to that, I think there's still value. that we have to keep talking to more and more doctors to justify that, but kind of depends on how people's thinking is on this. I think with people getting more worried about resistance and overprescribing of antibiotics, just being able to differentiate and identify the species gives you some useful information. But that's our limitation and the trade-off for the technologies. But yeah, we get things like that. Some people are skeptical. No one has, luckily, and they shouldn't, have come up and say this is just wrong, it's not going to work. We've done enough research to be 100% confident this isn't a problem. If we came up with something, then it's time to close up shop and move away. But there are competing things that take other approaches, and we've gotten the questions like, well, PCR is moving very quickly, maybe they can catch up to you. Like, well, maybe.
What's PCR?
Polymerase chain reaction. So the other way to do rapid identification is through genetic analysis of a sample. You can get an answer in about an hour. But it's much more complicated of a process. You have to collect your sample, break the cells open, add reagents, put it in a pretty large instrument, and wait the hour to get the results. And it gives you a breakdown based on what DNA is present, what bacteria is present in the sample. So that's partly our competition, but they haven't moved yet to point of care. It's a much more expensive test. So right now, those tests are running just for the materials alone about $150. It doesn't work well for wounds because wounds always have some bacteria in them. They're not sterile. So you would get a ton of false positives. So it's good for blood testing where you're not supposed to have any bacteria. And if you see something, you know that you need to treat them. People don't use this sort of test for wound care because you'd get positive. Same thing with pneumonia and sputum samples. Your lungs have bacteria in them. You would get positives all the time. Ours, since you can quantify the molecular things, you can set a threshold and say you're only interested above this level and for these molecules and how it's changing. So that's a competing technology. So we always have to explain how we differentiate ourselves on that. But that's
Other applications or funding opportunities to explore the food industry for foodborne pathogens or homeland security, because they do a lot of behind-the-scenes biotourism monitoring and stuff.
I did look at the food industry. Food industry's tough. The margins are very, very low. So I talked to a couple startups that are in this space looking at food diagnostics. And they're working away at it and speeding up the process. It's strange to think about it. It's actually, the sensitivity requirements are even higher than for testing in biologic and human samples, because the standard is that one bacteria in like a head of lettuce that's been homogenized in a blender. And our technology would probably still need some incubation time before we would see a signal. And they want something for like $1 a test. And I don't have experience in the food industry, so I just did not want to go down that route. There's people that are going down that route. Maybe down the road they might license something from us and put it into that sort of a platform, but right now that's almost more of a personal choice. We might be able to even get funding had we gone down that route, but we don't have expertise in that.
What do you project the cost for your test?
So we have a cost of what we think the cost of goods is going to be, and then it's a question of how much is the consumer going to pay for it. So we think we can make our product, because it's going to be a single-use disposable, eventually for about $1 or $2 a piece for a panel. And our sensor is very similar to a glucose meter, and we can essentially give that away when people just buy a pack of sensors. But we will charge a lot more than that. So because right now, if you go to like a veterinarian, cost is about $100 to $150, and you're waiting 2 to 3 days. So that's going to be a business decision. Same thing on the human side, what those costs are going to look like. On the human side, you also have to see what Medicare is going to reimburse for these tests and things like that. So that's the business side. So I don't want to say, oh, it's pennies and we're going to make it super cheap, but that's one of these interesting trade-offs. So it's definitely economically viable, and we've talked to potential manufacturers, and they can make these sorts of test strips for us. We can definitely make the box, and that. So in terms of costs for making it, I think we're in pretty good shape. Yeah, last question.
Thank you. I think it's great that you're going down that, that startup route. But I could easily imagine you were at a point, at some point, when you think, like, I could go out on my own, or I could knock on, like, another big fish in this industry, in that space is iStat. Like,
Yeah.
what was your choice? Like, why did you go it alone versus knocking on iStat's door to just give them one more tool in their huge group?
So,
And you can, like, buy you out.
no, great question. Maybe someday they will. I actually haven't knocked on the right person's door yet at iStat, but we have talked to other companies. They're not just going to write a check yet, so they want to see some more data. They're willing to partner with us, so strategic partnerships and things like this, and maybe they'll buy us out at some point.
The more you scale, the more expensive you're going to be later.
Yeah, so it's a trade-off on their part. We'll see.
Cool.
Yeah, great.
Thank you so much. Thanks so much for coming. Again, we can continue the conversation, and you can pepper him with your questions out by the food and drinks. Just a heads up, for those of you who don't know, UNH actually has their own I-Corps site grant, and we are going to be doing another round of training in March. So, if you have an idea that you're looking to explore, just reach out to any of us at UNHInnovation. There's also an application on UNHInnovation's site, and for the external people, we're looking for mentors for that as well. All right, thank you so much.
Thank you.